Hello, my name is Napa Pakhirat, number 22. Today I'm going to talk about chromosome genetic material and genetic inheritance. So I'm gonna start with chapter 4, chromosome and genetic material. Molecular inheritance. DNA, the substance of inheritance, the most celebrated molecules of our time. Hereditary information encoded in the chemical language of DNA and reproduced in all cells of your body. Next, evidence that DNA can transform bacteria. Experiment. Bacteria of the S or smooth strand of Streptococcus pneumonia are pathogenic. Bacteria of R or rough strain like a capsule and are non-pathogenic. And this is the result of the experiment. If you put living S cell into the mouse will die. And if you put living R cells into the mouse is still alive. And also heat kill S cell. If you put it in, mouse is still alive. If you put mixture of heat kill S cell and living R cell, the mouse will die. Evidence that viral DNA can program cell. Viruses that infect bacteriophage, are used as tool by researchers in molecular genetics. Additional evidence that DNA is the genetic material. It was already known that DNA is a polymer of nucleotide, a nitrogenous base, a sugar and phosphate group. Erwin Chargaff analyzed the base composition of DNA. The, the evidence of molecular diversity among species made DNA a more credible candidate for the genetic material. Next, building a structural model of DNA. Scientific inquiry. Wasn't and Craig deduced that DNA was a double helix through observation of the X-ray crystallographic. Wasn't and Craig reasoned that there must be additional specificity of pairing dictated by the structure of the bases. Each base pair form a different number of H bonds. A and T form 2 bonds and G and C form 3 bonds. Like this. Maurice Wilkin and Rosalind Franklin were using our X-ray crystallography to study molecular structure. Franklin included that DNA was composed of two antiparallel sugar phosphate backbones with the nitrogenous base pair in the molecule's interior. The nitrogenous bases are paired in specific combinations A, T, C, and G. Many proteins work together in DNA replication and repair. Relationship between structure and function is manifest in the double helix. The two strands of DNA are complementary. Each strand acts as a template for building a new strand in replication. So, at the first picture, the parent molecule has two complementary strands of DNA. Each base is paired by hydrogen bonding with its specific partner A with T and G with C. The first step in replication is separation of the two DNA, and then each parental strand now serves as a template that determines the order of nucleotide along a new complementary strand. Next, the nucleotides are connected to form the sugar phosphate backbone of the new strand. Each daughter DNA molecule consists of one parental strand and one new strand. DNA replication is semi-conservative. Each of the two new daughter molecules will have one old strand derived from the parent molecule and one newly made strand. The parent molecule invents and two new daughter strands and builds based on base pairing rules. So, first, conservative models. The two parent strands reassociate after acting as template for new strands, thus restoring the parental double helix. And next, semi-conservative model. The two strands of the parental molecule separate and each function as a template of synthesis of a new complementary strand. And the last one, dispersive models. Each strand of both daughter molecules contains a mixture of old and newly synthesized DNA. Michelson and Stahl experiment. This experiment supported the semi-conservation models of DNA replication. So this is the experiment. First, bacteria cultured in medium containing, and then bacteria transferred to medium containing, and then DNA sample contribute after 20 minutes after first replication, and last, DNA sample centrifuged after 40 minutes after second replication. So, conclusion of this experiment. Mesalon and Stahl conclude that DNA replication follow the semi-conservative model by comparing their result to the result predicted by each of the three models in the figure. The first replication in the 14N medium produced a band of hybrid 15N to 14N DNA. This result eliminated the conservation model. A second replication produced both light and hybrid DNA, a result that eliminated the dispersive model and supported the semi-conservation model. So this is the first replication and second replication. So getting started, origin of replication. A eukaryotic chromosome may have hundreds or even thousands of replication origin. So first, replication begins at a specific state where the two parental strands separate and form replication bubbles. Second, the bubble expands laterally as DNA replication proceeds in two directions. And third, eventually the replication bubble fills and synthesis of the daughter strand is complete. In eukaryote, DNA replication begins at many stages along the giant DNA molecules of each chromosome. And in this micrograph, three replication bubbles are visible along the DNA of a cultured Chinese hamster cell. Antiparallel elongation, DNA polymerase add nucleotide only to the free, free end of a glowing strand along one template strand of DNA. The leading strand DNA polymerase III can synthesize a complementary strand moving toward the replication fork to elongate the other new strand of DNA. The lagging strand DNA polymerase III must work in the direction away from the replication fork. Antiparallel elongation, synthesis of leading and lagging strand during DNA replication. First, DNA polymerase III elongates DNA strand only in the 5' to 3' direction, and then one new strand, the leading strand, can elongate continuously 5' to 3' as the replication fork progresses, and then the other new strand, the lagging strand, must go in an overall 5' to 3' to 5' direction by addition of short segment Okazaki fragment that go 5' to 3'. And last one, DNA ligases join Okazaki fragment by forming a bond between their free ends. This results in a continuous strand. Priming DNA synthesis. DNA polymerase can't initiate the synthesis of a polynucleotide. The intito nucleotide strand is an RNA or DNA primer. Only one primer is needed for synthesis of the leading strand, but for synthesis of the lagging strand, each Okazaki fragment must be primed separately. So first, primers join RNA nucleotide into a primer. Then DNA polymerase III adds DNA nucleotide to the primer, forming an Okazaki fragment. Then after reaching the next RNA primer, DNA polymerase III falls off, and after the second fragment is primed, DNA polymerase III adds DNA nucleotide until it reaches the first primer and fall off. Then DNA polymerase I replaces the RNA with DNA, adding to the 3' end of fragment 2. DNA ligase form a bond between the newest DNA and the adjacent DNA of fragment 1. And finally, the lagging strand is the region is now complete. Other proteins that assess DNA replication, helicase, topoisomerase, Single-strand binding proteins are all proteins that assist DNA replication. So this is the meaning of the protein. It is like the meaning, the function of leading strand and lagging strand, function of leading strand and function of lagging strand. A summary of DNA replication. First, helicase unwind the parental double helix. Second, molecules of single-strand binding protein stabilize the unwound template strand. Third, the lagging strand is synthesized continuously in the 5' to 3' direction by DNA polymerase III. Then fourth, primase began synthesis of RNA primer of fifth Okazaki fragment. Fifth, DNA polymerase III is completing synthesis of the fourth fragment when it reaches the RNA primer on the third fragment. It will dissociate, move to the replication fork, and add DNA nucleotide to the 3' end of the fifth fragment primer. 6. DNA polymerase I remove the primer from the 5' end of the second fragment, replacing it with DNA nucleotide that it adds one by one to the 3' end of the third fragment. The replacement of the last RNA nucleotide with DNA leaves the sugar phosphate backbone with a free 3' end. And then 7, the last one, DNA ligases bond the 3' end of the second fragment to the 5' end of the first fragment. The DNA replication machine as a stationary complex. The various proteins that participate in DNA replication form a single large complex. A DNA replication or called machine. The DNA replication machine is probably stationary during the replication process, replicating the end of DNA molecules. The end of eukaryotic chromosome DNA gets shorter with each round of replication. Proofreading and repairing DNA. DNA polymerase proofread newly made DNA, replacing any incorrect nucleotide. In mismatch repair of DNA repair. Enzyme correct error in base pairing. The nucleotide excision repair enzyme cut out and replace damaged stretches of DNA. First, a thymine dimer distorts the DNA molecules. Second, a nuclease enzyme cuts the damaged DNA strand at 2 points and the damaged section is removed nucleus. Then third, repair synthesis by a DNA polymerase fills in the missing nucleotide. And fourth, last one, DNA ligases seal the free ends of the new DNA to the old DNA, making the strand complete. Replicating the end of DNA molecules. Eukaryotic Mo- Chromosomal DNA molecules have at their ends nucleotide sequence telomeres. If chromosome of germ cell becomes shorter in every cell cycle, essential genes would eventually be missing from the gamete they produce. An enzyme called telomerase catalyzes the lething of telomeres in germ cells. A chromosome consists of a DNA molecules packed together with proteins. Chromatin undergoes changes in packing during the cell cycle through interphase. Chromosomes aren't highly condensed. Most chromatin is loosely packed in the nucleus during interphase and condenses prior to mitosis. Loosely packed chromatin is called chromatin. During interphase, a few regions of chromatin are highly condensed into heterochromatin. Dense packing of the heterochromatin makes it difficult for the cells to express genetic information coded in this region. Histone can undergo chemical modification that result in changes in chromatin organization. In a bacterium, the DNA is called supercoiled and found in a region of the cell called the nucleus. Chromatin, a complex of DNA and protein, is found in the nucleus of eukaryotic cells. Next, gene expression. The process by which DNA directs protein synthesis includes two stages. Number one, transcription from DNA to RNA. Number two, translation from mRNA to protein. Evidence from the study of metabolic defects. Archibald Garand was first suggested genes decade phenotyper through enzyme. Linking genes to enzyme required understanding that cells synthesis and degrade molecules in a series of steps, a metabolic pathway. Nutritional nutritional mutants in Neuropara Scientific Inquiry. George Bendel and Edward Tatum exposed bread mold to X-rays, creating mutants that were unable to survive on minimal medium. Using crosses, they identified three classes of arginine deficient mutants, each lacking and different enzyme necessary for synthesizing arginine. So this is the experiment. So next, the products of gene expression, a developing story. Some proteins aren't enzyme So researchers later revised the hypothesis: one gene, one protein. Many proteins are composed of several polypeptides, each of which has its own gene. Basic principles of transcription and translation. RNA is the intermediately between genes and the protein of which they code. Transcription: it is the synthesis of RNA under the direction of DNA. It produces mRNA. And translation: it is synthesis of polypeptide which occurs under the direction of mRNA. Ribosome: it is the state of translation. A primary translation is the in situ RNA transcription from any genes. The central dogma: it is the concept that cells are governed by a cellular chain of command. It's DNA to RNA and to protein. Like this. So the first one is in bacteria cell or prokaryotic cell, and the second one is in eukaryotic cells. Codons: the triplets of bases. The flow information from genes to protein is based on a triplet code, a series of non-overlapping 3 nucleotide words. These triplets are the smallest unit of uniform length that can code for all the amino acids. During transcription, one of the two DNA strands called the template strand provides a template for ordering the sequence of nucleotide in an RNA. Transcription. And then during translation, the mRNA-based triplet codons, called codons, are read in the 5' to 3' direction. Each codon specifies the amino acid to be placed at the corresponding position along a polypeptide. Codon along an mRNA molecules are read by translation machinery in the 5' to 3' direction. Each codon specifies the addition of one of 20 amino acids. Cracking the code. Of the 64 triplets, 61 codes of amino acid. 3 triplets are stop signal to end translation. The genetic code is redundant but no ambiguous. Codons must be read in the correct reading frame in order for the specified polypeptides to be produced. Like this. Evolution of the genetic code. The genetic code is nearly universal, shared by the simplest bacteria to the most complex animal. Genes can be transcribed and translated after being transplanted from one species to another. Transcription is the DNA direct synthesis of RNA. A closer look. Transcription, the first stage of gene expression, can be examined in more detail. RNA synthesis is catalyzed by DNA polymerase, which pries the DNA strands apart and hook together the RNA nucleotide. The DNA sequence where RNA polymerase attached is called the promoter in bacteria. The sequence signaling the end of transcription is the terminator. The strand of DNA that is transcribed is transcription unit. Synthesis of an RNA transcription. There are three stages of transcription. First, initiation, second, elongation, and third, termination. So this is the picture of transcription stage in RNA. So you can see that promoter is here, and the first one it will initiation first, and then it will go to elongation, and last one is termination. RNA polymerase binding and initiation of transcription. Promoters signal the initiation of RNA synthesis. Transcription factors mediate the binding of RNA polymerase and the initiation of transcription. A promoter called a Tata box is crucial in forming the initiation complex in eukaryotes. So first, in the picture, a eukaryotic promoter include a Tata box, and then several transcription factors must be bind to the DNA before RNA polymerase II can do so. And then additional transcription factor bind to the DNA along with RNA polymerase II, forming the transcription initiation complex. And then next, elongation of the RNA strand. As RNA polymerase move along with the DNA, its untwisted double helix 10 to 20 bases at a time. Transcription process progressed at a rate of 40 nucleotides per second in eukaryotic. A gene can be transcribed simultaneously by several RNA polymerase. Termination of transcription. The mechanism of termination are different in bacteria and eukaryotes. In bacteria, the polymerase stopped transcription at the end of the terminator, but in eukaryotes, the polymerase continues transcription after the pre-mRNA is cleaved from the growing RNA chain. The polymerase eventually falls off the DNA. Eukaryotic cell modify RNA after transcription. Enzyme in the eukaryotic nucleus modify pre-mRNA before the genetic message are dispatched to the cytoplasm. During RNA processing, both ends of the primary transcript are usually altered. RNA splicing. These non-coding regions are intervening sequences or intron, and the other region are exon. RNA splicing remove introns and join exon, creating an mRNA with a continuous coding sequence. Alteration of mRNA ends. Each end of a pmRNA molecule is modified in a particular way. The 5' end receives a modified nucleotide 5' cap. The 3' end gets a poly A tail. These modifications share several functions. They seem to facilitate the export of mRNA. They protect mRNA from hydrolytic enzyme. They help ribosome attach to the 5' end. In some cases, RNA splicing is carried out by spiciosome consists of a variety of protein and several small nuclear ribonucleoprotein or SNRNPS. So they will cut out intron like this. So this is RNA transcription. The functional and evolutionary importance of intron. Such variations are called alternative RNA splicing. Because of alternative splicing, the number of different proteins an organism can produce is much greater than its number of genes. Proteins often have a molecular architecture consisting of discrete regions called domains. In many cases, different exon codes of the different domains in a protein. Exon snuffing by result in the evolution of new proteins. The functional and evolutionary importance of intron. Such variations are called alternative RNA splicing. Because of alternative splicing, the number of different proteins an organism can produce is much greater than its number of genes. Proteins often have a modular architecture consisting of discrete regions called domains. In many cases, different exon codes for the different domains in a protein. Exon shuffling by result in the evolution of new protein. Translation is the RNA-directed synthesis of a polypeptide. A closer look. Molecular components of translation. A cell translates an mRNA message into protein with the help of tRNA. Molecules of tRNA are not identical. Each carries a specific amino acid on one end. Each has an anticodon on the other end. Ribosome association and initiation of translation. The initiation stage of translation brings together mRNA and tRNA with the first amino acid and the two ribosomal subunits. First, a small ribosomal subunit binds with mRNA and a special initiator of tRNA. Then the small subunit moves along the mRNA until it reaches the start codon or AUG. Protein's initiation factor bring in the large subunit that completes the translation initiation complex. The structure and function of transfer RNA. A tRNA molecule consists of a single RNA strand that is only about 80 nucleotides long. Accurate translation requires two steps. The first step, a correct match between the tRNA and an amino acid done by the enzyme amino acetyl tRNA synthetase. And second step, a correct match between the tRNA anticodon and an mRNA codon. Flexible pairing at the third base of a codon is called wobble and allows some tRNAs to bind to more than one codon. Elongation of the polypeptide chain. During the elongation stage, amino acids are added one by to the preceding amino acid. Each addition involves protein called elongation factor and occurs in three steps. First, codon recombination. Second, peptide bond formation. And third, translocation. Building a polypeptide, the three stages of translation: first indentation, second elongation, and third termination. All three stages require protein factor that aid to the translation process. Next, ribosome. Ribosome facilitates specific coupling of tRNA anticodon with mRNA codon in protein synthesis. A ribosome has three binding states for tRNA. First, the P state. It holds the tRNA that carry the going polypeptide chain. And second, the A stage holds the tRNA that carry the next amino acid to be added to the chain. And third, E state the exit state where discharged tRNAs leaves the ribosome. Termination of translation. Termination occurs when a stop codon in the mRNA reaches the A state of the ribosome. The A stage accepts a protein called a release factor. The release factor causes the addition of a water molecule instead of an amino acid. The reaction releases the polypeptide and the translation assembly then comes apart. So you can see that in this, it's release factor at stop codon and then they free polypeptide. Polyribosome. A number of ribosomes can translate a single mRNA simultaneously, forming a polyribosome. Polyribosome enable a cell to make copies of a polypeptide very quickly. So first, it's incoming ribosomal subunit at first of mRNA, and then the glowing polypeptide and complete polypeptide, and this is the end of mRNA. Completing and targeting the functional protein. Often translation is not sufficient to make a functional protein. Polypeptide chains are modified after translation. Complete protein are targeted to specified stage in the cell. Protein folding and post-translational modification. During and after synthesis, a polypeptide chain spontaneously coils and folds into its three-dimensional shape. Protein may also require post-translational modification before doing their job. Other polypeptides come together to form the subunit of a protein. Targeting polypeptide to a specific location. Two populations of ribosome are evident in cell: free ribosome and bound ribosome. Free ribosome mostly synthesize protein that function in the cytosol. Bound ribosome makes protein of the endomembrane system and proteins that are secreted from the cell. Polypeptide synthesis always begins in the cytosol. Polypeptide distinct for the ER or for silation and marked by signal peptide. A signal recognition particle binds the signal peptide. The SRP brings the signal peptide and its ribosome to the ER. Types of point mutation. Point mutation within a gene can be divided into into two general categories. First, base pair substitution, and second, base pair insertion or deletion. Substitution. A base pair substitution replaces one nucleotide and its partner with another pair of nucleotides. Missense mutation still code for an amino acid, but not necessarily the right amino acid. Nonsense mutation change an amino acid codon into a stop codon, nearly always leading to a non-functional protein. Insertion and deletion. Insertion and deletion are addition or lossence of nucleotide pair in a gene. These mutations have a dissociation effect on the resulting more often than substitution do. The insertion or deletion if nucleotide may alter the reading frame, grounding a frame shift mutation. Next, mutagens. Spontaneous mutation can occur during DNA replication, recombination, or repair. Mutagens are physical or chemical agents that can cause mutation. While genes expression differs among the domain of life, comparing genes expression in bacteria, archaea, and eukaryotes. Bacteria and archaea differ in their RNA polymerase, termination of transcription, and ribosome. Archaea tends to resemble eukaryotes in this respect. In eukaryotes, the transcription and translation are separated by the nuclear envelope. In archaea, transcription and translation are likely coupled. And the last one, what is a gene? Revisiting the question, we have considered genes as a discrete unit of inheritance, a region of specific nucleotide sequence in a chromosome, a DNA sequence that codes for a specific polypeptide change. In a summary, a gene can be defined as a region of DNA that can be expressed to produce a final functional product that either a polypeptide or an RNA molecule. Chapter 5: Genetic Inheritance. Mendel's experimental quantitative approach. Advantages of pea plants for genetic study. There are many varieties with distinct heritable features or character. For example, like flower colors, character variants. For example, purple or white are called traits. Melting of plants can be controlled. Each pea plant has sperm-producing organs and egg-producing organs. Cross-pollination can be achieved by dusting one plant with pollen from another. In a typical experiment, Mendel made two contrasting true breeding varieties, a process called hybridization, The true breeding parents are the P generation. The hybrid offspring of the P generation are called the F1 generation. When F1 individual self-pollinate, the F2 generation is produced. The law of situation. When Mendel constructing true breeding whites and purple flower peat plants, all of the F1 hybrid were purple. When Mendel crossed the F1 hybrid, many of the F2 plants had purple flowers, but some had white. Mendel discovered a ratio of about 3 to 1 3 to 1 purple to white flower in the F2 generation. Mendel called the purple flower a dominant trait and the white flower color a recessive trait. Mendel observed the same pattern of inheritance in 6 other pea plants character, each represented by 2 traits. So this is the experiment. Mendel's model. Mendel's developed a hypothesis to explain the 3 to 1 inheritance pattern he observed in F2 offspring. The first concept is that alternative version of genes account for variation in inheritance character. These alternative versions of a gene are now called allele. The second concept is that for each character, an organism inherits 2 alleles from one from each parent. The 2 alleles at a locus on a chromosome may be identical. The third concept is that if 2 alleles at a locus differ, then the dominant allele determines the organism appearance and the other has no noticeable effect on appearance. The fourth concept, how known as the law of saturation stage, that the 2 alleles for a heritable character separate during gamete formation and end up in different gametes. The possible combination of sperms and egg can be shown using Punnett square and diagram for predicting the result of genetic cross between individuals of known genetic makeup. A capital letter represents a dominant allele, and the lowercase letter represents a recessive allele. Next, useful genetic vocabulary. Homozygous, an organism with 2 identical alleles for a character. Heterozygous, an organism that has 2 different alleles for a gene. Phenotype, physical appearance like white or purple, and genotype, genetic makeup. For example, it's like P and small p. Monohybrid cause, a cause between such heterozygous. Test cross. Genetic experiments determine the genotype of an organism if any offspring display the recessive phenotype. The mystery parents must be heterozygous. The law of independent assortment. Mendel developed the law of Saturation by following a single character. The F1 offspring produced in this class were monohybrid, individuals that are heterozygous for one character. Mendel identified his second law of inheritance by following two characters at the same time. Crossing two true breeding parents differing in two characters produce F1 in the F1 generation. Using a dihybrid cause, Mendel developed the law of independent assortment. The law of independent assortment states that each pair of alleles surgerate independently of each other pairs of allele during gamete information. Genes located near each other on the same chromosome tends to be inherited together. The law of probability governed Mendelian inheritance. Mendel's law of saturation and independent assortment reflects the rules of probability. In the same way, the allele of one gene saturate into gametes independently of another gene's alleles. Solving complex genetic problems with the rules of probability. A dihybrid or other multi-character cross is equivalent to two or more independent monohybrid crosses occurring simultaneously. In calculating the chance of various genotypes, each character is considered separately, and then the individual probabilities are multiplied together. The multiplication and addition rules apply to monohybrid crosses. The multiplication rule states that the probability that two or more independent events will occur together is the product of their individual probability. Saturation in a heterozygous plant is like flipping a coin. Each gamete has a chance of carrying the dominant allele and a chance of carrying the recessive allele. The rules of addition can be used to figure out the probability that an F2 plant from a monohybrid cause will be heterozygous rather than homozygous. Degrees of domains. Complete domains occur when the phenotype of the heterozygote and domain heterozygotes are identical. In incomplete domains, the phenotype of F1 hybrid is somewhere between the phenotypes of the two parental varieties. In condominants, the domains allele affect the phenotype in separate, distinguishable ways. Extending Mendelian genetic for a single gene. Inheritance of character by a single gene may depend from simple Mendelian patterns in the following situation, like when alleles aren't completely dominant or recessive, and when a gene has more than two alleles, and the last one, when a gene produces multiple phenotypes. The relation between domains and phenotype. The domains of allele can vary depending on the level of analysis. Organismal level. In Tay-Sachs disease, the allele is recessive. The disease occurs only in homozygous individuals. Biochemical level, the phenotype is incompletely dominant. Heterozygotes show intermediate enzyme activity. And molecular level, the alleles are condominant. Both normal and defective enzymes are produced in heterozygotes. Frequency of dominant allele. Dominant alleles are not necessarily more common in population than recessive alleles. For example, one baby out of 400 in the USA is born with extra finger or toes. The allele for the unusual traits is dominant to the allele of the more common traits of 5 digits per appendage. For example, the recessive allele is far more prevalent than the population's dominant allele. Multiple allele. Most genes exist in population in more than 2 allelic forms. For example, the 4 phenotypes of the AOB blood group in humans are determined by 3 alleles for the enzyme or I that attaches A or B carbohydrates to red blood cells. The enzyme encoded by the enzyme A allele adds the A carbohydrate, whereas the enzyme encoded by the IB allele adds the B carbohydrate. The enzyme encoded by the I allele adds neither. Pleiotrope. Most genes have multiple phenotypic effect property. Pleiotropically, for example, phenotypic alleles are responsible for the multiple symptoms of certain hereditary diseases such as cystic fibrosis and sickle cell disease. Extending Mendelian genetic for 2 or more genes. In epistasis, a gene at one locus alters the phenotypic expression of a gene at a second locus. For example, in mice and many others, mammal coat color depends on two genes. One gene determines the pigment color and the other genes determine whether the pigment will be deposited in the hair. Polygenic inheritance. Quantitative characters are those that vary in the population along a continuum. Quantitative variation usually indicates polygenic inheritance and additive effect of 2 or more genes on the single phenotype. Skin color in humans is an example of polygenic inheritance. Nature and nurture the environmental impact on phenotype. The norms of reaction is the phenotypic range of a genotype influenced by environment. For example, hydrangea flower of the same phenotype range from blue violet to pink depending on soil acidity. Norms of reaction are generally bordered for polygenic character. Such characters are called multifactorial because genetic or environmental factors collectively influence phenotype. Integration Mendelian views of heredity and variation. An organism phenotype includes its physical appearance, internal anatomy, physiology, and behavior. An organism's phenotype reflects its overall genotype and unique environmental history. Many human traits follow Mendelian patterns of inheritance. Humans are not good subjects for genetic research. Generation time is so long, like it have to use 2 or 3 years. Parents produce relatively few offspring, and breeding experiments are unacceptable. However, basic Mendelian genetics endures as the foundation of human genetics. Pedigree analysis. It is a family tree that describes the interrelationship of parents and children across generations. Inheritance pattern of particular traits can be treated and described using pedigree. Pedigree can also be used to make prediction about future offspring. So this is a picture of pedigree. Recessively inherited disorder. Many genetic disorders are inherited in recessive manner. The behavior of recessive allele. Recessively inherited disorder shows up only in individual homologous for the allele. Carriers are heterozygous individuals who carry the recessive allele but are phenotypically normal. Albinism. Albinism is a recessive condition causing lack of pigmentation, while the change of two carrier or aware allele melting is typically low. Consanguineous mating increase like this, like livelihood. Many societies discourage close relative marriage to reduce the risk of receive diseases. Consequence, mating increased the chance of mating between two carriers of the same rare allele. Most societies and cultures have laws or taboos against marriage between close relatives. Cystic the most common lethal genetic disease in the United States, striking one out of every 2,500 people of European descent. The cystic allele results in defective or absent chloride transport channel in plasma membrane. Symptoms include mucus buildup in some internal organs and abnormal absorption of nutrients in the small intestine. Sickle cell disease affects 1 out of 400 African American. The disease is caused by the substitution of a single amino acid in the hemoglobin protein in red blood cell. Symptoms include physical weakness, pain, organ damage, and even paralysis. Dominantly inherited disorder. Some human disorders are caused by dominant allele. Dominant allele that cause a lethal disease are rare and arise by mutation. Achondroplasia is a form of dwarfism caused by a rare dominant allele. Huntington's disease, a degenerative disease of the nervous system. The disease has no obvious phenotypic effect until the individual is about 35 to 40 years of age. Multifactorial disorder. Many diseases such as heart disease and cancer have both genetic and environmental components. Little is understood about the genetic contribution to most multifactorial diseases. Genetic testing and counseling. Genetic counselor help prospective parents assess the risk of passing on genetic disorder using family histories and probability rules. Amniocentesis, tasting the fluid surrounding the fetus. embryonic well sampling, testing a sample of the placenta, ultrasound and fetuscopy, visual assessment of fetal health in utero, newborn screening. Some genetic disorders can be detected at birth by simple tests that are now routinely performed in most hospitals in the United States.